In the last part of lecture 15, we want to talk a little bit more about urine formation, some of the forces involved in that. We'll talk about a very important number called glomerular filtration rate and what controls that. This is once again from chapter 25 in Merib. Let's begin by talking about some of the forces involved in glomerular filtration. Just a reminder, glomerular filtration is the movement of the capillary plasma across the filtration membrane out into Bowman's capsule or the glomerular capsule, if you remember that from before. Very similar to what we talked about when we talked about systemic capillaries. In the glomerulus, we have forces that govern whether or not fluid is going to move out of the capillaries or back into the capillaries. This is just a little bit more complicated than what we talked about with the systemic capillaries because we have one additional force that we'll talk about in a second. Remember when we talked about systemic capillaries, we talked about net filtration pressure. Basically, this was the algebraic sum of the forces moving fluid out of the capillaries and the forces moving fluid back into the capillaries. When we talked about systemic capillaries, we talked about hydrostatic pressure of the blood. This was the force that tended to push fluid out of the capillaries. And we also talked about the colloid osmotic pressure, or simply the osmotic pressure, that tended to pull fluid back into the capillaries. Now, when we talk about the kidneys, we have one additional complicating factor, and that's something called the capsular hydrostatic pressure, as you see down here. One of the things we didn't have to consider with the systemic capillaries is what happened to that fluid after it left the vessels. In other words, there was no boundary to constrain the fluid that seeped out of the capillaries. That fluid would eventually be pulled back into the capillaries. However, when we talk about the glomeruli and about the capsules of the kidney, remember that we have an epithelial cup that is basically catching the fluid that comes out of the glomerular capillaries. This small confined space that's in here, called the capsular space, is where fluid is going to accumulate. Because the space is so small, what this gives rise to is something that we refer to as capsular hydrostatic pressure, and this force tends to push fluid back into the glomerular capillaries. So we actually have three forces that we have to think about when we talk about glomerular filtration or about movement of plasma and fluids across the capillary membranes out into the capsular space. So the net filtration, as you see up here, is the algebraic sum of the glomerular capillary hydrostatic pressure, which is here, minus the forces that oppose that movement, which are first the capsular hydrostatic pressure that we just talked about, as well as the glomerular capillary osmotic pressure. And the net filtration normally is positive. That is, it favors fluid movement out of the glomerular capillaries. It normally is this way. Now, one of the things we need to talk about is about pressures here. Remember that we defined before the pressure inside the glomerulus as being about 55 to 60 millimeters of mercury. So we said before that the pressure in the glomerular capillaries is higher than what we saw in the systemic capillaries. And I think you'll appreciate now that the reason we need such a high glomerular hydrostatic pressure is to overcome the additional force that we have involved of the capsular hydrostatic pressure, which tends to move fluid back in. So let's take a look at these forces. As we said, the glomerular hydrostatic pressure is around 60 millimeters of mercury. We express that as a positive number, meaning that it's going to push fluid out. So this direction we represent as positive. Now that's opposed by two different forces. One is the plasma colloid osmotic pressure, or simply the osmotic pressure that's present in the blood. And this is the force that tends to pull fluids back into the glomerular capillaries. And then as we just talked about, we have that one additional complicating force, which is the capsular hydrostatic pressure. This also tends to move fluid back toward the glomerulus, so we would represent this as a negative force. When we sum these up, we find out that we have about 10 millimeters of mercury net filtration pressure, or what we represent as NFP. So this is the net force tending to move fluid out of the glomerular capillaries. This is not a great margin. It's only 10 millimeters of mercury, so consequently, if the pressure in the glomeruli drops, the net filtration pressure becomes less very, very quickly. So one of the primary things, as we'll see in a couple of minutes, that controls glomerular filtration is in fact the glomerular hydrostatic pressure, or the blood pressure within the capillaries of the kidney. Now the amount of fluid that moves out from the glomerular capillaries into the capsular space every minute is known as the glomerular filtration rate. We define this formally as the amount of fluid that's pushed into the glomerular capsules of all the nephrons of both kidneys every minute as being the glomerular filtration rate. Very important number for you to remember. The average glomerular filtration rate is about 125 milliliters per minute. Let's summarize what we just talked about and add a couple of more points to that. First of all, remember that net filtration pressure, although it's normally positive, is relatively low. That is, we're talking about a net filtration pressure of only 10 millimeters of mercury. The glomerular hydrostatic pressure is the blood pressure in the glomerular capillaries. Remember that this is typically higher than in other systemic capillaries. And so this is around 60 millimeters of mercury or so, and that high pressure is required because of the capsular hydrostatic pressure, tending to push fluid and water back into the capillaries. Now, anything that affects filtration pressures will also affect glomerular filtration rate. And as we mentioned before, the blood pressure really is the most important factor that will change the glomerular hydrostatic pressure and thus change the net filtration pressure. Now, as it says on the bottom of this slide, a mean arterial pressure fall of about 10% at the glomerulus itself will severely impair glomerular filtration. What does this mean? Well, think about the normal pressures at the glomerulus. That pressure of 60 millimeters of mercury, if we were to take a 10% drop in that, that would reduce that to about 54 millimeters of mercury. So it would reduce that hydrostatic pressure by 6 millimeters of mercury. That would reduce the net filtration pressure by 6 millimeters of mercury as well. And now we'd have a net filtration pressure of only 4 millimeters of mercury, tending to push fluid out. You can imagine then that if we had a fall of 15 to 20% of the glomerular hydrostatic pressure, this would certainly stop any filtration. In fact, it would cause fluid to move back into the glomeruli of the kidney. Now, as we'll see in a couple of minutes, those big falls in blood pressure are not typical of the glomeruli because they have their own mechanisms for ensuring that the blood pressure remains fairly high. But they can only do that within certain limits. So if the mean arterial pressure throughout the body starts to decline and it falls very rapidly and falls by a large amount, then the glomerular filtration pressures at the kidney will fall. So we'll take a look at those in a little while, but the glomerulus does regulate its own pressure and tries to keep the pressure within very narrow limits. So once again, the glomerular filtration rate is directly proportional to net filtration pressure. The higher the net filtration pressure, the more glomerular filtrate we make. The lower the pressure, the less we make. This is a very important number for you to remember. Once again, the glomerular filtration rate is about 125 milliliters per minute, and that gives us our 180 liters of filtrate per day. So what this basically means is that all of the plasma is filtered about 60 times every day. I'll let you calculate that on your own and figure out how we got that number. The other thing that we want to mention is that urine output is only about 0.6 to 2.5 liters per day, or we say an average of about 1.8 liters. What this means, as you see on the left, is that the total glomerular filtrate every day is 180 liters. So 180 liters of fluid is pushed out by all the glomeruli in both kidneys every day. However, what we release as urine is on average only about 1.8 liters. This tells us that about 99% of all the filtrate gets reabsorbed or claimed back to the body in a day. And we're going to talk about how that's done a little bit later. Now, how do we control glomerular filtration rate? Remember, we said that there were sympathetic nerves that followed the blood vessels into the kidney, and these actually will affect the diameters of the afferent and efferent arterioles. So let's take a look at how the diameter of the afferent and efferent arterioles affects glomerular filtration. Let's just draw a simplified diagram here and imagine that we have a situation like this, where here is the afferent arteriole, here would be the efferent arteriole. In the middle, of course, we would have the glomerular capillaries. Now I want you to imagine what would happen if we increase the radius of the afferent arteriole. In other words, we push this out a little bit and we had a situation like this, and the radius of the efferent arteriole didn't change at all. Well, we're bringing more fluid in now because we've increased the radius of the afferent arteriole. However, we're squeezing all that extra fluid into the same space. What do you think is going to happen to the pressure here? Sure, the pressure is going to increase. And if the pressure increases, the net filtration pressure increases. That means more filtration at the kidney, and that means an increased GFR. So as you can see here, the radius of the afferent arteriole is proportional to GFR. We say that if the radius goes up, GFR goes up. If, on the other hand, the radius goes down and we send less fluid into the glomerulus, that means we decrease GFR. Now let's take a look at what happens when we change the diameter of the efferent arterioles. Here once again we have the situation where we have the afferent arteriole here, the efferent arteriole here, in the middle we would have the glomerulus. What would happen if we increase the radius of the efferent arteriole? So let's say now we made the radius this instead of what it was before. Notice that the fluid that we're sending in here via the afferent arteriole doesn't have much of a constriction to go through if we increase the size of the efferent arteriole. What is that going to do to the pressure in the glomerulus? That's going to reduce it. So we have less of a difference between the size of the afferent and efferent arteriole when the efferent arteriole expands. That's going to decrease the net filtration pressure, and that's going to cause a decrease in GFR. So notice that the radius of the efferent arteriole is inversely proportional to GFR. If the radius increases, GFR decreases. If the radius decreases, GFR increases. This is a summary table of the different factors that affect GFR. We just talked about the changes in the afferent and efferent arteriole, those you see here. So if we have an increased amount of sympathetic stimulation, that means a vasoconstriction. And that means a decreased radius here and a decreased radius here. Notice the effect of this. A decreased radius of the afferent arteriole leads to a decrease in GFR. However, a decrease in the radius of the efferent arteriole leads to an increase in GFR. And the opposite is true if we have a vasodilation occur. Remember that the radius of the afferent arteriole is directly proportional to GFR. The radius of the efferent arteriole is inversely proportional to GFR. And down the bottom of the table you can see the three factors that are important in controlling fluid movement across the glomerular filtration membrane. This is the capillary hydrostatic pressure. Remember that this is the only force that tends to move fluid out of the glomerular capillaries. And then we have the colloid osmotic pressure, and we also have the capsular hydrostatic pressure. Both of these tend to move fluid back in. And so you see the relationships here. If we increase glomerular capillary hydrostatic pressure, we increase GFR. These other two are inversely related to GFR. So if we increase the colloid osmotic pressure, we decrease GFR, or if we increase the capsular hydrostatic pressure, we also decrease GFR. These relationships are important for you to understand. Now, there are two mechanisms of regulation at the kidney. Similar to what we talked about before, we have one mechanism which is called autoregulation. Remember, this happens at the level of the tissue itself without involving the nervous system. And then, of course, we have regulation by the nervous system. So we're going to talk about both. Let's first consider autoregulation at the kidney. And as you can see, this maintains glomerular filtration rate despite large changes in local blood pressure at the kidney. Somewhere between 80 or 90 to 180 millimeters of mercury is the range in which the kidneys can still maintain the same GFR, and this is quite a considerable range. So consequently, normal day-to-day fluctuations in blood pressure don't really cause large changes in water and solute excretion or changes in GFR. The only problem comes when the blood pressure goes too low, for example, hypovolemic shock, where we're losing blood. When the blood pressure gets lower than about 80, then glomerular filtration will suffer. Let's talk about exactly how the kidneys carry out the autoregulation. As you see here, there are two mechanisms. The first one is known as a myogenic mechanism. The myogenic mechanism actually reflects a property of the vascular smooth muscle that's in the afferent arteriole. It contracts when it's stretched, and it relaxes when it isn't stretched. So rising systemic blood pressure stretches the vascular smooth muscle in the arteriolar walls, causing the afferent arterioles to constrict. This constriction then restricts the blood flow into the glomerulus and prevents glomerular blood pressure from rising to high levels, which might damage the glomerulus itself. Declining systemic blood pressure, on the other hand, causes dilation of the afferent arterioles and raises glomerular hydrostatic pressure. Both of these responses help to maintain normal net filtration pressure and normal GFR. The second mechanism that we want to consider is what's called a tubuloglomerular mechanism. This is really directed by the macula densa cells in the juxtaglomerular apparatus that we looked at earlier. When GFR increases, there is not enough time for reabsorption, and the concentration of sodium chloride in the filtrate remains high. So the macula densa cells respond to high levels of sodium chloride in the filtrate by releasing vasoconstrictor chemicals. This would be the renin that we're going to talk about in a little while, that cause intense constriction of the afferent arteriole that reduces blood flow into the glomerulus. So this drop in blood flow decreases the net filtration pressure in GFR, and that in turn slows down the flow of filtrate and allows more time for the processing of sodium chloride. On the other hand, when there's low sodium chloride concentration, this causes vasodilation of the afferent arterioles. This allows more blood to flow into the glomerulus, and that's going to increase the net filtration pressure and the GFR. So this is called the tubuloglomerular mechanism. So these are two mechanisms by which the kidneys carry out their autoregulatory functions. Now let's talk just a little bit about neural or autonomic regulation. Remember, we said that sympathetic postganglionic fibers follow the blood vessels into the kidneys and can cause a very intense vasoconstriction of afferent arterioles. As we said before, remember the radius of the afferent arterioles is directly proportional to GFR. So if we vasoconstrict the afferent arterioles, reduce the amount of blood that goes into the glomerulus, that's going to reduce the GFR. This helps to conserve water and also redirects blood flow to other organs. Part, as you can imagine, of the sympathetic fight or flight response. We said in AMP1 that during that fight or flight response, we want to restrict blood flow to kidneys, to the digestive system. Instead, we'd like to redirect blood flow to muscles, to heart, and those sort of things that actually need it during that emergency situation. This also stimulates the juxtaglomerular apparatus to secrete renin. We're going to talk about the renin-angiotensin system in just a minute. This may override autoregulatory mechanisms that we just talked about. So in other words, if there's a large change in the body or something's going on with the body, and the kidney is carrying out its autoregulatory mechanism at the same time, the nervous system signals can override the normal autoregulatory response. Now, hormonal system regulation is really by the renin-angiotensin system. This is going to stabilize blood pressure and extracellular fluid volume. We're going to talk about that in just a second. Remember, in the endocrine system, we talked about something called atrial natriuretic peptide that's secreted by the heart. What this does is to basically get rid of more fluid in the body. So the signal, remember, for atrial natriuretic peptide to be secreted was an increase in blood pressure. And what we need to do in that case is to increase GFR, increase fluid loss from the body. So this would be dilating the afferent arteriole, constricting the efferent arteriole. So we're basically going from a situation where we would have something like this, as we drew out before, to a situation where we're dilating the afferent arteriole, making that bigger, and making the efferent arteriole even smaller. So much more of a constriction here, as you can see, so the pressure inside the glomerulus would definitely go up. This would increase GFR. This would in turn increase fluid loss at the kidney. Now, the very last thing we want to talk about in this lecture is the renin-angiotensin system. We just want to review this. We're going to flesh this out just a little bit more than we did previously. There's a couple of other things we want to talk about. Remember, in the event that blood pressure declines, what happens is the kidneys are signaled to produce renin. Renin is an enzyme. And what renin does is act on a precursor molecule in the blood that's made by the liver called angiotensinogen. This converts angiotensinogen into angiotensin 1, which is an inactive form. And then in the lung capillaries, for the most part, we have another enzyme called angiotensin converting enzyme that will convert angiotensin 1 to angiotensin 2. So angiotensin 2 is actually the active portion of this whole sequence of events. And what I would like you to know is what exactly angiotensin 2 does, and that's shown in this little box right here. One of the things angiotensin 2 does is to cause vasoconstriction throughout the body. This will certainly raise blood pressure, as we know from the cardiovascular system. The other thing angiotensin 2 will do is increase aldosterone secretion. This will promote sodium uptake at the kidney. And as we said earlier, if we move sodium from one place to another, water is going to follow along. So the net effect of this is that when we secrete aldosterone, we increase salt and water reabsorption. The other thing angiotensin 2 will do is increase ADH secretion. And remember, ADH is basically a water-preserving type of hormone, so it decreases the amount of water we lose from the body, thus antidiuretic hormone, as we said before. So when the posterior pituitary secretes this, this reduces the amount of water loss from the body, thus increasing the plasma volume and increasing blood pressure. And finally, angiotensin 2 will also increase thirst. And this is the only way that we can get more water into the body is by taking it in from the outside. So these are all conserving mechanisms, and this is the only way that we can get more water into the body to increase blood pressure. Now, the signals for the release of renin in the first place, as we said, a decline of blood pressure, that one you see here. We can also, in some cases, have sympathetic stimulation of the juxtaglomerular apparatus, as we talked about a second ago. This can also cause an increase in blood pressure and an increase in secretion of renin. And finally, when we have a decline in the osmotic concentration of tubular fluid, the macula densa will detect that and will also cause an increase in renin secretion. Okay, so that finishes up this part 3 of lecture 15. I'll see you very shortly for lecture 16, and we'll talk a little bit more about the kidney. See you then.